अस्सलामुअलैकुम नाज़रीन, आज का हमारा नया टॉपिक है नर्व इम्पल्स। नर्व इम्पल्स इज़ अ वेव ऑफ electrochemical changes which travel along the length of neuron involving chemical reaction and movement of ion across the cell membrane. The electric potential is a measure of capacity to do electrical work in the represent the type of storage energy which is due to the separation of charges. Charge are separating barrier in the plasma membrane. The electric potential that exists across a cell membrane known as membrane potential. The typical neuron at rest is more positive electrically outside than inside the cell membrane. The net difference is charge between the inner and outer surface of non-conducting neuron is called resting membrane potential. The major factors which are involved in resting membrane potential: the sodium and potassium ion of many kinds of ion present in nerve cell and surrounding fluid. Sodium Na plus and potassium K positive ion are the most important. Sodium ion are 10-fold higher in concentration out than inside the membrane surface, where the potassium ion are 20 times more concentration inside the out. All the neurons have very active sodium and potassium pumps located in their cell membrane. Driven by the splitting of ATP, these pumps transport Na positive out and K positive into the cell, both against their respective concentration gradient. For every 2 potassium pumps, they are actually transported inward. 3 Na positive are pulled outward. So, inside become more negative than the outside of cell membrane of neuron. The negative ions, organic ions, the large negative organic ions such as protein and organic acid etc. are much more inside the membrane that outside, where they are only in negligible concentration. This makes the inside of neuron membrane more negative. Leakage of potassium ion from neuron. The cell membrane is virtually impermeable to all ion except K positive and potassium. Since the membrane is slightly permeable to K positive of it leak out of cell. The loss of this positive ion from the neuron by diffusion accounts of more negative charge inside than outside the cell membrane of neuron. The conduction of nerve impulse. Diagram. Initially, the nerve impulse under the normal condition, the nerve impulse is inhibited by a appendicular stimuli called thyroid stimuli applied at one end of neuron, and its result is remarked localized change in the resting membrane potential. It disappears for a brief instant and is replaced by a new potential called action or active membrane potential, which is in the form of impulse. During this stage, the inner membrane surface become more negative than outside. This change is so brief, so spare a millisecond, then that only a portion of neuron is in the active membrane potential state. The major factor involving in changing the resting membrane potential of active site. The fixed 17.4 रेस्टिंग पोटेंशियल। इसी के साथ हमें दीजिए इजाज़त। इंशाअल्लाह हम आपको मिलेंगे अगली वीडियो में। अल्लाह निगहबान। अल्लाह हाफिज़। अपना ख्याल रखिएगा। अल्लाह हाफिज़।